Bismillahirrahmanirrahim. Welcome back to AmreenaMehrajTutorials.
In the name of Allah. Assalamu alaikum, my dear students. May Allah subhanahu wa ta'ala shower his blessings upon everyone who is watching this video. My dear students, this is today lecture number 3, which is for class 12th as well as for entrance students, and we are studying genetics. I hope you remember. Out of genetics we have started mutations, and out of mutations we have completed gene mutations, if you remember. Now chromosomal mutations we also started, and out of chromosomal mutations we have already completed genomic mutations. Now we are supposed to complete this topic, which is chromosomal aberrations. Inshallah, we'll complete this topic today. Before beginning this lecture, I would like to convey you people that these are certain contact details which will be helpful for the students to drop their queries, because I was being queried a lot by MBBS students, so I am not available on Facebook Messenger anymore. I have uninstalled that app because of certain reasons. Now I want those students to not to worry at all. I'll be available at multiple platforms. You can mail your queries on my email. I have provided 2 emails. One is mehrajamreena@gmail.com. Another is amtutorials100@gmail.com. You just feel free to drop your queries or mail me these queries on these 2 particular emails. You will find me easily on Facebook. I'm on my Facebook page is AmreenaMehrajTutorials, and my Facebook account is already known to you. That was AmreenaMehraj. That is functional, but Messenger I have uninstalled. Instagram account I have, you can follow me on that also. This is am_tutorials100. The best place for you people to drop your queries is the YouTube comment box itself. Don't hesitate, just drop your queries. I'll be answering it as I was answering on Messenger, that Facebook Messenger. My dear students, this is very important. Please pay note of it. Don't forget to like, share and comment on my channel. Simultaneously subscribe to my YouTube channel, and you just press that particular bell icon if you want to get notified with my videos. Now let us start the topic itself. Okay, so as far as these aberrations are concerned, my dear students, chromosomal aberrations, this is the topic for the day. Chromosomal aberrations, again a type of mutation. But in this particular mutation, what you will see, my dear students, is what is basically happening in this case is that the chromosomes are undergoing some morphological change. Means their structure is changing. So suppose this is your normal chromosome, the general form of chromosome I'm drawing over here. So this chromosome is undergoing certain morphological changes. Let me write down over here. So in this particular chromosomal aberration, what is happening is a morphological change in what? In the structure of chromosome. In this particular category of mutation, what is happening to the chromosome? It is undergoing some morphological change. Very simple will be for you people to remember that what is happening is a kind of, what is it basically? A kind of structural change. The structure of chromosome is getting changed. Now the question is, how could the structure change? See, it has nothing to do with addition of another chromosome, maybe 1 or 2 or 3, or maybe an addition of a set, which we studied under genomic mutations. I am quoting about aneuploidy and euploidy. This is not that case. In this case, what will happen, that the structure itself will get changed. Maybe a particular segment of this chromosome will get deleted, means the chromosome will become shorter. Or what will happen is that a particular segment will be repeated multiple times. So there will be the scenario where there will be sometimes deletion of a segment happening, or sometimes there will be duplication of a segment happening. Generally, we must have this basic information before starting this topic, is that this particular region, this pinched region of chromosome is called what? It's called centromere. We must have this first-hand information first of all. And usually the short arm and long arm of a chromosome have specific names. Say for example, this arm, which is the short arm, it's called P arm. And this arm, which is say for example the long arm of the chromosome, it's called the Q arm. Okay, so these are some specific terms which we'll be using today. So this is for the short arm of the chromosome, and this is the term we use for the long arm of the chromosome. So usually if you will log on to Google and you search about some information about chromosome, you will always be getting some information written as 12q21, or you'll be written suppose like 3p22. These are the coding versions of representing any gene on a particular chromosome. So you just remember, see, you have these letters used in labeling a chromosome, P and Q. What are they basically? They are simply the different arms of a chromosome. P arm represents the short arm, Q arm represents the long arm of a chromosome. Similarly, the central region, which is present at different places in a chromosome. You have this information from class 10th that the centromere could be located at the center, it could be slightly away from center, either it could be absolutely at the terminal. Accordingly, you have metacentric, submetacentric, telocentric, acrocentric chromosomes. So those things are not so important, but this much is very important. You must have this information about this central region called centromere and two arms, P arm and Q arm, the short arm and the long arm of the chromosome. Now let us get back to the chromosomal aberrations. So in this case, you will see what? You will see sometimes a particular section of P arm is getting deleted. A particular section of Q arm is getting deleted. These are the things which are happening under chromosomal aberrations. So in certain cases, my dear students, you will see in these aberrations sometimes only one chromosome is involved, sometimes two chromosomes are involved. Accordingly, you have two types of chromosomal aberrations. One is called intrachromosomal aberrations. One type of chromosomal aberration is called intrachromosomal aberration, and another is called interchromosomal aberrations. Okay, so again you have two types of chromosomal aberrations: intrachromosomal aberrations and interchromosomal aberrations. Very simple. In this case, only one chromosome is involved. Simple. So whatever is happening, whatever is the sort of mutation which is happening, either in terms of deletion or whatever is the possibility, that is happening with one chromosome. Okay, in this case you have the involvement of you have the involvement of two chromosomes. Means there you will see in this category that a minimum two chromosomes are involved in this particular type of chromosomal aberrations. Remember one thing, whether it's intrachromosomal aberration, whether it's interchromosomal aberrations, both of them are going to have deleterious effects on an organism, and these are usually the cause of various genetic disorders. Remember this point. Okay. Now, under intrachromosomal aberrations, we have to discuss two types. First one is going to be deletion. Okay. So number one type of chromosomal aberration is deletion. Number second type which we'll discuss is inversion. Okay. Number third type will come under interchromosomal. So this again has two types. So the first type under interchromosomal aberration will be duplication. Okay. And the another type under interchromosomal will be translocation. Okay. So So in total, how many we are supposed to study under chromosomal aberrations? Number 1, deletion. Number 2, inversion. Number 3 is duplication. Number 4 is translocation. Remember, these two under intrachromosomal and these two under interchromosomal. Once I will discuss them one by one, you will come and understand why in these two cases only one chromosome is involved and why in these two cases two chromosomes are involved. Now let us take them one by one. So we will be discussing deletion. That will be followed by inversion, that will be followed by duplication, that will be followed by translocation. Let us take deletion first of all. Okay, so I'll be taking up number 1. Number 1 is deletion. Very interesting. My dear students, as the name indicates, what will be happening in deletion? There will be loss of a particular segment of a chromosome. Let me write down first of all. Loss of a segment of chromosome. You can define it in different terms also. You can say loss of some genes from a chromosome. You can say loss of a fragment of a chromosome. Or you can say simply deletion or loss of a segment of chromosome. Now pay attention. Over here, suppose this is a chromosome I am drawing. Okay. I'll try to show you a chromosome like this. Okay. Now, upon this chromosome, I'll place a sequence of genes. Suppose I will code them like A gene, B gene, C gene. Similarly, we have D, E, and F. So, this is a chromosome. I've taken one chromosome. Pay attention. One chromosome is over here and you have a sequence of genes on it. Okay. Now, my dear students, I'm telling you that what happened during the formation or maybe during the process of cell division, what happened is that suppose this particular segment got deleted. This particular segment got deleted. This could happen maybe once you will expose the cell to the radiations. Maybe if you are exposing a cell to X radiations, this can happen. That means breakage of a chromosome will take place. Breakage of the genetic material will take place. Once this particular segment will be broken down, what will be the resultant chromosome? How will the resultant chromosome look like? It will be something like this. Pay attention. It will be something like this. Okay, I'll try to make it shorter so that you will get this idea. Okay, it will be something like this now. Because A is lost, you have B and C, D, E, and F. What has happened in this case is deletion of a segment of chromosome. Simple. Or, my dear students, now I will tell you that just go and delete this particular part of the chromosome. Delete this particular part of the chromosome. Pay attention. Now I have deleted this particular section. Okay. The result will be something like this again. Result will be like this. It will be like A is there, B is there, C has been lost, D is there, E is there, F is there. These are the representation of the genes. Okay. Now this is also deletion. This is also deletion. Your chromosome has become shortened. Now, the difference is this type of deletion is called, it's called what? It's called terminal deletion. If you can guess why is it called terminal deletion. This type of deletion is called intercalary deletion. If you can guess why. Very simple. Absolutely correct, all of you. Terminal deletion because there is a loss of segment from the terminal position. See, you have lost a segment, you have lost a portion of the chromosome from its end and is called terminal position. That's why you call this deletion terminal deletion. In this case, there is loss of middle segment of a chromosome. Very simple. That's why you use the word, whenever middle portion is concerned, it's called intercalary. Okay, so you have two types of deletion. One is called terminal deletion, another is called intercalary deletion. Okay, so now you got to understand why deletion comes under intrachromosomal aberration, because in this case only one chromosome is involved. Okay, this is the reason why you call it intrachromosomal aberration. Now there are different, deleterious effects which could appear in an organism if something like this will happen in a particular chromosome. I told you earlier that these things can result in various genetic disorders. Let me give you some examples over here. Those examples are very important as far as NEET is concerned. Let me give you some examples in which the same thing is happening. Either terminal deletion is happening or intercalary deletion is happening. So let us get some examples. There is a genetic disorder which we will be discussing in detail once I will take up genetic disorders. The name of the disorder is Cry-Du-Chat. Cry-Du-Chat is a genetic disorder in which the child, once it cries, the cry of a child is similar to a cat cry. Okay, under this genetic disorder, what is wrong basically with the child is that there is a deletion in a particular chromosome. Actually the half of short arm, I have told you earlier what short arm is. Half of the short arm of chromosome number 5 in this case gets deleted. So half of the portion of the short arm of this particular chromosome gets deleted. The resultant genetic disorder is Cry-Du-Chat. You also call it cat cry in simple terms. Okay, Cry-Du-Chat is a French term. Similar is the case with another genetic disorder. That the name of that genetic disorder is it's called Prader-Willi syndrome. What is the condition in Prader-Willi syndrome is that there is a deletion on long arm of chromosome number 15. So there is a deletion on what? Long arm of chromosome number 15. It results in this syndrome which is called Prader-Willi syndrome. The peculiar feature of Prader-Willi syndrome is the person suffers mental retardation. There are multiple other effects of this particular disorder. Okay, another example goes like this. You have a well-known example of this disease which is called Williams syndrome. In case of Williams syndrome, there is deletion of a particular portion. So let me write down a portion of long arm of chromosome number 7 gets deleted. Okay, I'm writing a portion. So you have this term P arm or Q arm. Either say long arm or short arm. Okay, so there is one more example. One is that's called Jacobson's syndrome. In Jacobson's syndrome, what happens is that a portion of long arm of chromosome number 11 gets deleted. So see, concerned chromosomes are getting deleted. A particular portion of them is getting deleted. The result is either Cry-Du-Chat, Prader-Willi syndrome, Williams syndrome, Jacobson syndrome. Each syndrome has a range of effects. Okay. Another example goes like Wolff-Hirschhorn. Here it's called HIRS. Hirschhorn. HHORN. Hirschhorn syndrome. It's also a syndrome. In this case, you will see that the short arm of chromosome number 4 gets deleted. Okay. Short arm of chromosome number 4 gets deleted. That means this arm gets completely deleted. See, in this case, you had half of the short arm getting deleted. Here you have the short arm of chromosome 4 getting deleted. The resultant syndrome is Wolff-Hirschhorn syndrome. Remember these examples, they are very important as far as examinations are concerned, specifically your competitive examinations. Now, let us go and understand about the second type of intrachromosomal aberration, that is your inversion. Let us now start inversion. So the second type is inversion. Inversion is again, number one point about inversion is it's again intrachromosomal aberration. So just one chromosome is involved again. Now, what is going to happen in case of inversion is very, peculiar. See, in case of inversion, you have a particular segment of chromosome getting, first of all, deleted. Then that segment undergoes inversion at an angle of 180 degree. Then it gets joined back with the chromosome. So how can we define it? We can write down that in this case there is, orientation of a segment is reversed by 180 degrees. So we write down orientation of a, specifically we'll write down deleted segment changes by 180 degrees. See, let me make you understand what is happening. We will take a chromosome again. Suppose this is a normal chromosome I'm taking over here. The sequence of the genes is like this: A, B, C, D, E, F, G, H, I, J. This is the normal chromosome. And A, B, C, D, E, F, G, H, I, J represent the various genes which are located on this particular chromosome. Now, when inversion has to happen, first what will happen is a particular section of the chromosome will get, first of all, deleted. Okay, deletion will happen first. Then that particular segment will undergo rotation by an angle of 180 degree. That will totally be reversed. And then that segment joins back with the chromosome. That particular step is called inversion. So what will we do? First of all, we will do deletion. Once you will get a segment out of this DNA, out of this particular chromosome, then that segment has to undergo what? It has to undergo rotation by how many degrees? 180 degrees. That's why we use the word inversion. You have to invert it, okay? Then it will join back. Say for example, if this thing happens over here, suppose this particular segment undergoes inversion, then what will be the scenario? I'll show it over here. Scenario will be like this. A and B first of all will get deleted. Then there will be 180 degree rotation in this manner. B will come first, followed by A. Rest will be same. So see, inversion of AB segment took place. AB got deleted first, then inverted, BA, then join back. B, it will be followed by C, D, E, F, G, H, I, and J. Notice what has happened to this region. It has undergone inversion. Okay. In another scenario, let me take this particular portion. Let me take this particular segment of this chromosome, which I have shaded with green color. Suppose this region undergoes inversion. Again, first of all, it will get deleted. Then it will rotate by 180 degree. Then it will join the chromosome back. The situation will be something like this. The resultant I will show over here. So this will be the condition. In that case, you will have a condition like this. I'm showing for this particular CD region. AB will be as such. Okay. Now what has happened to CD? Inverted. You will have D followed by C. Then there will be E. Then F, G, H, I, J. See? Notice this particular section. BC. Okay. Similarly, my dear students, now I will take this section. E and F. Okay. I'll do the same inversion process with this particular fragment or this segment of the chromosome. What will be the result? I will show over here. Suppose I will show over here. Okay. This is the third case. Okay. The scenario will be AB is as such. CD is as such. Now you have E and F getting inverted. What will be the scenario? You will have, let me place them a bit far from each other, C, D. Then you have, it was E over here. Then now you will have F here and E here. Rest is same, G, H, I, J. G, H, I, J. What is happening in all these cases is inversion. But can you find out the difference? The difference is at which position this inversion is taking place. In this case, inversion is happening at the end. What we will use for this case, we'll call it terminal inversion, if I am correct. Because the inversion or the change in the orientation of deleted segment by 180 degree takes place at the end of the chromosome. This is a case where inversion is taking place in the middle section. This is the case where inversion is taking place in the middle section. So both of them are the examples of what? They are the examples of intercalary inversion, as was the case for deletions. Okay, intercalary inversion, because the middle section is involved. This is also intercalary inversion. Okay, now is there something else we can notice over here? Yes, you can notice this particular region. You can notice this. What is this basically? This is the centromere. I have told you earlier, okay? A chromosome has got this particular region, which is the highly condensed region. This is the centromere, okay? So this over here is the representation of the centromere. See, in this case, centromere is not involved. Whereas in this inversion, which is shaded in blue, in this case, centromere is involved. So accordingly, this type of inversion in which centromere is not involved, it's not included in inversion, it's called paracentric intercalary inversion. So let me write down the full form of this case is paracentric, because centromere is not involved. In this case, centromere is involved. We will call it pericentric inversion. So remember, in paracentric inversion, in this case, centromere is not involved. Simple. In this case, why is it called pericentric? Because centromere is involved. This is very simple. Now, practically, if it will happen in the chromosome, suppose this condition arises in chromosome, it will again cause a genetic disorder. If you will analyze this disorder, let me give you example over here. If you will analyze hemophilia, inshallah, we will be discussing its types, everything. I will be giving you detail about A, B, C type of hemophilia, everything we will cover under genetic disorders. But over here, remember, hemophilia A is a genetic disorder whose cause lies in these particular inversions happening to the gene which is coding for blood clotting factor 8. So this over here simply represent that hemophilia A is caused due to inversion mutation. This you can remember. Even inversion is responsible for various types of genetic polymorphisms. Okay? Genetic polymorphism arises. This genetic polymorphism arises in a particular population because of inversions. Okay, genetic polymorphism is basically nothing. It is two or more discontinuous genotypes because of either you say discontinuous genotypes or you say discontinuous alleles present in a population. Those difference in alleles arise because of inversions at the genetic level. So you can remember that genetic polymorphism, the best best example of genetic polymorphism which arises in Drosophila. The reason behind that is again inversion mutations. Remember, genetic polymorphism and genetic disorders like hemophilia A are caused due to inversion mutations. This was again an intrachromosomal aberration. Now let us take up the interchromosomal aberration. In that we will first of all discuss the type which is called duplication. Duplication. Duplication is a type of interchromosomal aberration. First of all, it's an interchromosomal aberration. So in this case, you will see involvement of two chromosomes. In this case, you will see what is participating is a homologous pair. A homologous pair of chromosome is participating. In humans, if we talk about humans, we have 23 such pairs. Chromosome number 1, it's a homologous pair. It has two members. One is coming from the maternal side, one has come from the paternal side. So each homologous pair is concerned over here. And now what is happening is basically, suppose if this is a pair of a chromosome, a homologous pair it is. So if this is chromosome number 1, this is also chromosome number 1. Then it forms chromosome number 1 homologous pair. So what is happening in this case is that this segment from one member is getting deleted, and then the same segment is going to go and it's going to join the other member of the pair. So it's going to come over here. So this is going to happen in this case of duplication. How we can put down this in words is that we can write down over here, in this case, a segment of DNA, or you can say the chromosome itself, is getting, first of all, what? First of all, it has to get, is getting deleted, okay? Number 1, it has to undergo deletion. And then it attaches to the other chromosome of the homologous pair. Okay, now let me make you understand. Say for example, over here you have a condition like this. Suppose I'll make a condition like this. I'll show you, I'll be drawing a homologous pair for understanding purposes. So it's a pair. Now over here we are not concerned about only one chromosome, we have to show both the chromosomes. So this is a homologous pair, okay? So over here, whatever will be the genes here, same are going to be present in this. So A, B, C, D, E, F, G, H, the sequence of the genes. Here also A, B, C, D, E, F, G, H. Now what is going to happen, my dear students, when duplication happens is basically this particular segment. Say for example, first of all, it will undergo deletion. So first of all, it will get deleted. Then it will be, attached with the other member of the same homologous pair. That means it will come and it will join over here. What will be the result? The result will be, let me show you the result over here. Result will be that this chromosome will become shorter. The condition will be like this. So you will have a chromosome like this. It has become shorter, if you can observe. Because it lost A, B, it's left with only C and D. Then it has E, F, G and H. Okay. The other chromosome will become longer. Okay. So you have A, B, original AB and the duplicated AB. A, B, then you have C, D, then you have E, F, G, H. What has happened in this case? Observe. This was the original chromosome, original gene which was present in this chromosome, and this is the region which actually belonged to the other member of the homologous pair, but it has now shifted to this particular chromosome. This is the scenario of duplication. But remember, what is involved in this case is this homologous pair. So this was a homologous pair earlier. Still, this is a homologous pair. But the condition has now become faulty. The mutation has appeared. You will see such things happening in various genetic disorders. Let me give you some examples. There is a genetic disorder which is called Pallister. The name of the genetic disorder is Pallister-Killian syndrome. Okay, this is a syndrome in which the same thing is happening. Chromosome number 12 will get some extra portion. So in this case, which is involved is your chromosome number 12 is experiencing what? Duplication. The result is this syndrome is called Pallister-Killian syndrome. Let me give you another example. There is a disorder which is called Charcot-Marie Tooth disease. It's called Charcot-Marie M-A-R-I-E tooth disease type 1, because it has got multiple types. This disorder has got multiple types. So tooth, because in this case a person involuntarily grinds teeth, that's why it's called tooth disease. But it has multiple effects. Majorly in this case, what is being damaged is the myelin sheath. So you'll see a lot of degradation happening to the nervous system in this particular genetic disorder. The name of the disorder is Charcot-Marie Tooth disease type 1. In this case, duplication happens to the short arm of chromosome number 17, or you can simply remember that in this case chromosome number 17 undergoes the duplication. So I have given you some of the examples for duplication type of chromosomal aberration. Number 1, you can remember Pallister-Killian syndrome. Number 2, you can remember Charcot-Marie Tooth disease type 1. These are the peculiarities associated with the duplication. Very simple. Now we have to take the last type, that is our translocation. Let's go to translocation. Let us now take translocation. The last type, 4th, is translocation. It's similar to duplication. Now you will come and understand the difference. Again, this is interchromosomal aberration. Okay, 2 chromosomes involved. But the difference, the way it differs from duplication is basically, in this case, what is involved is non-homologous, pair of chromosomes. So in this case, again you have 2 chromosomes, but they are not going to be belonging to the same original homologous pair. One will be, chromosome number 1, another will be chromosome 2. If 1 and 2 is forming pairing, it's called non-homologous pairing. Okay. So here you have non-homologous pairs of chromosomes between which there will be some exchange of segments. Try and understand how we can define it. Is that you can write down, in this case, a segment breaks and then it attached to a non-homologous chromosome. So in this case, a segment of a chromosome breaks. So first of all, deletion, okay, and attaches to a what? There it was attaching in duplication, it was attaching to homologous pair, another member. Here it is attaching to, it attached to a non-homologous chromosome. Let me make you understand. Suppose I'll show you over here, I'll make some arrangement of non-homologous chromosomes in this section. Pay attention. So try and understand. Look, observe carefully and understand. Okay. This is one pair I've made. It's slightly tilted, but it's okay. Okay, these are schematic representations. Okay, and another pair I'll make over here. I'll make you understand why I'm drawing so many. See, in this particular case, the arrangement is like, you just guess, A, B, C, D, E, F, and here you have A, B, C, D, E, F. Is it a homologous pair or a non-homologous pair? It's homologous because the genes represent in this chromosome same is the arrangement and orientation of gene on another chromosome. So this is what? This is homologous pair. Okay, done. In this case, you have PQRSTU arrangement, PQRSTU arrangement. Okay, my dear students, again, see, same. This is also what? Homologous pair. Pay attention. Now, these two different homologous pairs are, interacting. Now, I'll make you understand why I wrote non-homologous chromosome then. See, suppose something like this is happening. Let us put this into consideration. This particular segment. Let us notice this segment. Let us notice this segment. This segment is getting deleted. So, a segment of chromosome breaks. This segment breaks. Then, it attaches. It attaches to what? It attaches to a non-homologous chromosome. It comes and it attaches over here. Because this pair and this pair, they are totally non-homologous. Okay? So, these two are homologous with respect to each other. These two are homologous with respect to each other. But if you compare this and this, they are non-homologous with respect to each other. If you compare this and this, they are non-homologous with respect to each other. So, if something like this will happen, this will break off, then it will get shifted over here. Okay, what will be the result? See, the result will be that you will be left with something like this. Try and understand. You will be left with something like this, and this will be normal like this. Try and be smart in understanding what I mean in understanding it you. So, and this will become like this. Let us understand it thoroughly. See, the condition is AB is gone. What are you left with? C, D, E, F. Okay? A, B, C, D, E, F. It's as such. A, B, C, D, E, F. So, this is the condition of pair number 1. Okay? Now, what has happened in this? After getting deleted, this segment came and it joined with this homologous pair, which is non-homologous to it. Okay, so it came over here. Now you have a condition like A, B, then you have PQR. Okay, PQR, STU. Okay, and this is same PQR, STU. What has happened? See, observe these two and observe these two. This is the scenario, this particular thing, which I will cover in green. This is the scenario after translocation. Okay? This type of translocation, where the segment got transferred to a non-homologous chromosome. This type of translocation is called simple translocation. It's also called non-reciprocal translocation, but I use the word simple translocation. Now, my dear students, something like this is happening. Once this segment comes and joins the non-homologous chromosome, and in place of this particular segment, PQ, is getting transferred back to the chromosome. Okay? So, what is happening? A reciprocal thing. A segment coming to non-homologous chromosome, this non-homologous chromosome donating its segment to the donor. So, the situation will be like this now. See, the situation will be like this. The lengths will remain same. Observe keenly. Just observe keenly. There's no hurry at all. Okay. See what is happening? So you have AB going over here, but in place of AB, it is receiving this segment PQ. So you'll have PQ followed by C. Then you have D, E, F. This is same. Nothing is happening to this. This is A, B, C, D, E, F. Notice this thing. Okay. Now, this has received AB. So over here, it is A, B, rest is same. R, S, T, U. Okay. And the other one is normal. P, Q, R, S, T, U. Notice this. Notice the other one. Notice this type of case. So let me keep it different. Wait, I'll try to keep it different. Notice this green one, blue one. Notice the case which I have, blocked out in blue color. This is also translocation, but a segment is deleted over here. It goes and joins some non-homologous chromosome. This also deletes a particular section that goes to this particular chromosome. So this is called reciprocal translocation. Okay, reciprocal. And this is, you can also call it accordingly, non-reciprocal translocation, or you can simply call it simple translocation. Everything is evident once you will observe the sequence. See, you have to put what into consideration? AB. In this case, AB is missing, it has gone over here. And in this case, other case, AB is replaced by PQ. So you'll see PQ over here and AB over here. So the places which you have to notice, I will underline those places. Okay, so this portion is missing and it has gone over here. This is the case of translocation. So a segment of chromosome deleting, and then it is attaching to a non-homologous chromosome, which I have explained to you people. Now let me give you two examples for this particular case. Let me give you two genetic disorders which can arise because of such translocations. Okay, they are very important. Very, very important they are. One among them is a case of leukemia, which is called chronic myelogenous leukemia. Okay, that is caused because of translocation. Okay, another is Burkitt's lymphoma. That's also caused due to translocation. You have another reasons for these disorders also. You have another mutations also, but in these patients, you will see that there is a particular translocation also happening. So, example will be for this type of translocation. I mean to say, once this translocation will happen, resultant will be some, genetic disorder. One such disorder is chronic myelogenous leukemia. It's a type of leukemia. Okay, and in this case, what happens basically, there is a translocation from chromosome number 22. A segment of chromosome number 22 is shifted to chromosome number 9. So, chromosome number 22 and chromosome number 9 undergo this process of translocation. Accordingly, chromosome number 22 is converted into a different chromosome, which is Philadelphia chromosome. It's called Philadelphia chromosome. Okay, my dear students. So, why is it called Philadelphia chromosome? Because it has been named in a city called Philadelphia, which is in Pennsylvania. So, Philadelphia chromosome is this chromosome in which translocation is happened at between chromosome number 22 and chromosome number 9 specifically. This disorder is also called, in short form, you can write down CML, chronic myelogenous leukemia. Okay, this was number one type of genetic disorder which can arise due to translocation. Another genetic disorder which can arise due to translocation is Burkitt's lymphoma. Burkitt, lymphoma is lymphoma. Let me write down first of all. In this case, you have translocation happening between chromosome number 8 and chromosome number 14. So, a translocation between chromosome number 8 and 14 result in Burkitt lymphoma. You have other mutations and other things responsible for the cause of these two particular disorders, but they are associated with translocations. In this case, translocation between these two, and in this case, translocation between these two chromosomes. This was all I had for you people in this video lecture today. So we completed chromosomal aberrations. We are done with our topic called mutations. Now inshallah in my next video lecture we'll be starting off with genetic disorders. So inshallah stay tuned for that video. That's going to be very much informative and useful. Meanwhile, if you have any queries regarding today's lecture or my previous lectures, my comment box is available. You can drop your worthy queries in my comment section below. And don't forget to like, share and comment. Simultaneously subscribe to my YouTube channel so that I can reach to more and more students. I just want to thank all my students whom I have taught at different institutions. I thank you all with an open heart for praising me so much. Thank you so much. Allah bless you all. And this was all for today. Have a nice day, and do remember me in your prayers. That's it. Fi aman Allah.